What's up everybody? So in this video we're going to be talking about photosynthesis. Now I have a quick question for you. If I were to put you on an island without any resources or food, what's most likely to happen to you in the next few days? You're most likely to die. Certainly I would die. Now if we did the same thing with a plant, somehow it would so easily grow. It would so easily survive. It can so easily thrive in these conditions. Now, why is that? Why can a plant survive and we cannot? Well, the reason is photosynthesis. A plant can synthesize their own food with light. So photo here means light. Now, we cannot do that. If we want to survive, we need to eat other things, right? Otherwise, we can't survive. Now, what is it about plants that allows them to do photosynthesis and we cannot? We cannot just go and sunbathe and make our own food. We need to get up and eat a pizza later, right? It's just how it works. So why is that? Well, the answer lies in the cells. So we know all living organisms are made up of cells. So plants, right? We know they're made up of a kind of cell called a plant cell. Now, that's where the difference lies. Me and you, we're made up of animal cells. So the difference between, one key difference between a plant cell and an animal cell is one of the organelles. So a plant cell has this key organelle that is called a chloroplast. So this organelle we do not have. Me and you do not have, but plants have it. Now this is the reason, this organelle is the reason why plants can do photosynthesis and we cannot. Okay, so I get it then. Okay, so we need these plants have these chloroplasts and therefore they can do it. But how does photosynthesis work then? Well, let's look at that. So when we look at a chloroplast, it's an organelle that is stacked with a lot of these little discs. Okay, normally it's very full with these discs. These discs are called thylakoids, and they're like this, disky. But for the sake of this diagram, I'm only going to put a few of them here. Okay, because I'm going to use this space to explain something later. But know that in reality, these chloroplasts are full of these thylakoids. Now, thylakoids have a lot of water in them, H2O, right? So oxygen bounded by two hydrogens. Now, how photosynthesis happens is during light, when there's light outside. So during the day, light energy. Light energy is going to be shown everywhere. It's shined everywhere, right? We experience it as heat. Now, a plant can take in this light energy, specifically the chloroplast, more specifically the thylakoid. So the thylakoid can take in this light energy and use it to split apart, break apart this water molecule into its constituents. We call that photolysis. Photo means light and lysis means to split. So using light energy to split apart water, that's photolysis. Okay, you can think of it like a wave in the ocean. If you get smacked by a big wave, I'm pretty sure you can break apart too. Okay, so these waves, these light energy are just little waves that we cannot see with our own eyes. Now these waves can come and smack and break apart this water into its little parts. Okay, why? Ask me, my man, why do we care about this? How does this allow a plant to survive? Well, we're not quite there yet. So we broke apart now this water using photolysis. Already, look, we formed something interesting. Now we have oxygen. We know this happens a lot. So we're building up all of these little hydrogens and a lot of these oxygens. Now these oxygens, when they combine together, two of them, we form O2. This is the kind of oxygen that we breathe, right? We breathe in. We know humans inhale oxygen and we exhale what? We exhale carbon dioxide. Okay, why? Because oxygen we inhale because we need oxygen to do cell respiration. So when we eat food, me and you, okay, we get in a lot of nutrients. For example, glucose can be one of them. And we want to break this glucose down because glucose isn't ATP. We need ATP to survive. Our cells need ATP. So cell respiration is going to use oxygen and glucose and oxygen and all that to break down glucose into ATP. And as a byproduct of cell respiration, we make carbon dioxide. So a waste product will be carbon dioxide. So we exhale that. So we inhale oxygen to do cell respiration to break down this glucose into ATP, and then we exhale carbon dioxide as a waste product. Okay, that's very important to understand. So that's already one cool thing that happened with this photolysis. But there's more. We have this hydrogen. These hydrogens are going to leave the thylakoid. So they're in these little discs, right? And they will kind of be like, okay, well, I'm done here. I'm going to leave. And they leave into the surrounding area. Okay, now So now we have hydrogens here, but how are we going to make glucose with just hydrogens? Because we know actually, in actuality, glucose is made up of three things. Carbon, right? A lot of carbon, 6 carbons. It's made up of a bunch of hydrogens, and it's made up of oxygen. So right now we only have hydrogen, so we cannot make glucose. So how are we going to make glucose? We're missing carbon and oxygen. Can you think of where on this page we can get carbon and oxygen? From carbon dioxide, right? Us humans or us animals exhale carbon dioxide as a waste product. But one man's trash is another man's treasure. So our trash, our carbon dioxide, is going to be used by a plant. They love this carbon dioxide. They need it to make glucose, okay? Whereas they got rid of oxygen, we need it, okay, to do cell respiration. Let me do something here. So now we have carbon dioxide here and hydrogens, and they will combine together in a process called carbon fixation. We're fixing it all together into a glucose. And that's it. That's how we did photosynthesis. So photosynthesis is this process, using light energy, using water to make glucose and oxygen, right? Very important. We're going to write the chemical equation now, and you'll understand it much better. But in a nutshell, we converted light energy into chemical energy, because this chemical energy, glucose, can now be used to make ATP. So this plant, it just made its own food, glucose, but now it can use that glucose and break it down to do cell respiration to make ATP so they can survive. So I want you to not get confused. Plants do photosynthesis, they make their own glucose, but they also do cell respiration. They also break down their own glucose that they made to make ATP so that they can survive. Okay, so don't get it confused. Plants don't only do photosynthesis. They do photosynthesis and cell respiration. They make their own food and they eat their own food. Whereas us humans and animals, we can't make our own food, so we need to go and eat it somewhere else. Then we can break it down using cell respiration. Okay, awesome. So now let's look at the equation. Most teachers love to show this equation at first, but I like to show it at the end because now it'll make a lot more sense. So this is, in a nutshell, photosynthesis. We need carbon dioxide, right here. It's the source of carbon and oxygen in our glucose. We need water. Why do we need water? Because water is going to be split apart into our hydrogens that we need and our oxygen. So what did we make during photosynthesis? A lot of oxygen, right? This oxygen is going to be released into the environment. And as a final product, we made what? Glucose. Awesome, right? And we also needed light energy. So when you write this equation, you can write carbon dioxide plus water plus light energy equals glucose and oxygen, okay? That's photosynthesis for you, so that's awesome. So now I want to clear some things up with you real quick before we move on to more detail. Okay, so you can see that organisms work together. The oxygen that plants release can be used by us to do cell respiration, and the carbon dioxide released by us during cell respiration can be used by plants to do photosynthesis, okay? It's a working together process. Okay, here's the definitions of the actual processes, photosynthesis and cell respiration. Now you need to know two other words. One is autotroph. So an organism, like a plant, that can make their own food is called an autotroph, because auto means automatic and troph means feed. They can by themselves, feed. They can feed by themselves. They make their own food. Okay? When these autotrophs use light to make their own food, we call them photoautotrophs. Now, an organism like me and you, we call ourselves heterotrophs because we feed on other things. Hetero means other, troph means feed. We feed on other things, okay? So we are heterotrophs. We are organisms that cannot produce our own food. Okay, so that's very important to understand. Know these definitions, know what the difference is between an autotroph and a heterotroph, and remember, a plant can do photosynthesis and cell respiration. They make their own food and they eat their own food. They don't just do photosynthesis, okay? And we would only do cell respiration. We need to eat our food from somewhere else. Awesome. So now let's look at something in a bit more detail. We need to look at the light idea in a bit more detail. And I think it's kind of interesting. So when we look at the sun, this is our source of light energy, right? The light, the photosynthesis. Now light is shown, and we know the sun shines white light, right? We cannot really see it, it's transparent. But if we take a prism and shine the sunlight through this prism, what will happen is this white light will become many colors. So what this prism really does is it splits apart the white light into all the colors that it's made of. So when we mix all of these lights together, it looks white. But when we separate them, we can see what they're actually made of. Okay, and we can see all these colors, they are different colors because they're different wavelengths. So the longer wavelengths ones appear as red to our eyes, whereas these middle wavelengths appear as green to our eyes, whereas these short wavelengths appear as blue or purple to our eyes. So the different wavelengths is what makes something appear a certain color. Okay, now, why am I telling you this? You'll see shortly. So when we look at all the waves on the planet, there's very, very teeny tiny waves, very, very short waves, and very long waves. Us as humans cannot see all these teeny, very, very short waves. They're called gamma rays and X-rays and things like that. And the very long waves are radio waves. These waves we cannot see. They're either too big or too small or too frequent for us to see. But we can see a small spectrum in all these waves called visible light. We call it visible light because we can actually see these waves as color. Now, when we look at this little visible light region, we can see that those waves that have very short wavelengths, like 400 nanometers, remember the word nanometer, on some multiple choice that like to put micrometer and confuse you, and then that's the wrong option, not the correct option. So remember, nanometer is the unit you need to remember. So the very short wavelengths, so 400 nanometers would be these wavelengths that we see as purple, and the longer wavelengths would be those as red, okay? Now, remember this, okay? You're not going to understand it that much because it's a chemistry idea, but remember that those with the long wavelengths are lower energy. L for long wavelengths, L for lower energy. L for long wavelengths, L for lower energy, okay? And these short wavelengths are higher energy. So L for long and lower energy, okay? Don't worry about why, I'm never going to ask you why, okay? Just remember that. Might be a multiple choice thing. It's not too important, just might be a multiple choice thing. Okay, so now, as I just said, this sun, it shines this white light, which is actually, remember, it's actually made up of all of these wavelengths, and together they appear white. Now, in our chloroplast, remember our chloroplast? That's what's going to absorb this sunlight. Our chloroplast is an organelle. Remember, there's two words that sound very similar here, and I don't want you to confuse them. The chloroplast is the organelle. Inside the chloroplast, so inside our chloroplast, specifically inside our thylakoid, remember, that's the place, the thylakoid is the place where the light is actually absorbed from. In the thylakoid, there's something called a photosynthetic pigment, okay, because this is a little thing that is the actual thing absorbing the light. This is the very area that's attracting the light. Okay, so let me go back here. So in our thylakoid, in here is a photosynthetic pigment that is attracting the light energy, so that the light energy comes in here and breaks the water. Now that little thing, okay, is called chlorophyll. So there are many kinds of photosynthetic pigments. The main one that we need to care about is chlorophyll. It's a kind of pigment, the main one, okay, inside our thylakoids, and that is the one that's going to specifically absorb light. Okay, now when light is shined at our cell, at our chloroplast, at our thylakoid, at our chlorophyll pigment, these photosynthetic pigments can do one of two things. They can either absorb a wavelength or reflect a wavelength, okay? So remember, some wavelengths are long, some wavelengths are short. So this pigment will absorb some of them and reflect some of them, or it can absorb all of them and reflect all of them, okay? Now chlorophyll is a kind of pigment. Okay, and this photosynthetic pigment absorbs everything basically except green. Okay, so it will absorb blue, red, all of those, but it will reflect green. Okay, so remember, we're talking about the photosynthetic pigment of chlorophyll. Different photosynthetic pigments absorb different wavelengths and reflect different wavelengths. But chlorophyll is going to absorb red and blue light very well, and I'm going to show you more on the next page. And it's going to reflect green, okay? So it's going to reflect green. It's going to come in here, all the white light with all the wavelengths. It's going to absorb basically all of them. And the red and blue lights are the best absorbed, okay? And then green light is reflected. So you can see here, green light is reflected. And that's why we see plants as green, because green is the only wavelength that is not used. It is reflected back. so this wavelength will come back into our eyes, and therefore we will see it as green. So the reason why we see it as green is because of the fact that it reflects green waves and absorbs every other kind of wave. Okay. So when we look at a microscope image, so you can see the reason why plants are actually green is because of the chloroplast. Because the chloroplasts are the area that contain the chlorophyll, okay? And specifically the thylakoid. Specifically the thylakoid will contain the chlorophyll. So you can see, if we look at this image, all of the chloroplasts here are the green things, okay? And they're the reason why our plants look green. One interesting thing I want to point out here is notice how a lot of the chloroplasts are near the side of the cell. You can see there's a lot of cells here. One here, one here, one here. Notice how a lot of the chloroplasts are squeezed against the side. The reason why is because we know a plant cell has a lot of these things here, okay? A lot of these things here. And they are basically filled with water, and they can get so huge that they take up the entire cell and press all of the little organelles by the side, okay? So that's why you can see this huge space here is filled with water, and the other organelles are forced to be squished by the side. Okay? So that's why when you see on a test, sometimes you'll be confused. Don't be confused. You know these ones, when they're pressed against the side, that they're just chloroplasts. Okay, great. So now we're going to look at the actual absorption spectrum. So this here is a little graph that shows what wavelengths are the best absorbed by a certain photosynthetic pigment. I told you there are many kinds of photosynthetic pigments. The one we care about most is chlorophyll. Okay, it's the most common. So when we look here, we can see the wavelengths 400 nanometers to 700 nanometers. So this would be red, this would be blue, and then there's a whole gradient. And this will be absorbance, so how much they're absorbed. So we can see chlorophyll, this pigment, chlorophyll A and chlorophyll B, they're both chlorophylls, they absorb pretty well in the bluish area, bluish purple area, and pretty well in the red area, but they absorb very little in the green area. So all of the wavelengths that are here, red, orangey, are absorbed well, and the ones that are bluish purplish are absorbed very well, which is why I said here red and blue light is well absorbed by this chlorophyll pigment. So this will be, the red and blue light is what will be used to split apart that water to do the photolysis. Green is reflected, it is not used at all, okay? It is not at all used to do photolysis. So that's important. So we call this the absorption spectrum. This spectrum shows us which wavelengths are well absorbed by this pigment. So chlorophyll A and chlorophyll B, these two pigments absorb well these wavelengths and these wavelengths. They don't absorb well green wavelengths. Now there's another kind of photosynthetic pigment called carotenoids. They are present, there's a bunch of different carotenoids, and don't worry about them much, these are not the common ones. But this kind of photosynthetic pigment absorbs well this area here, and really doesn't absorb well red, orange, yellow at all. So basically, this kind of, whatever plant contains a lot of carotenoids, like the majority are carotenoids, they will appear orange. So if we look here, for example, a carrot, it contains a lot of these pigments. A lot of these pigments called carotenoids. So it appears orange because none of these are absorbed. They're all reflected. They're not really used. Only these here are absorbed. Okay, so that's very important. So make sure you understand that different plants have a different combination of these photosynthetic pigments. The most common is chlorophyll, so it's the most abundant. The other ones are there, but they're not really very concentrated. There's not a lot of them, so they don't really make an effect. So even though this plant here is green, that doesn't mean it only has chlorophyll pigment. It could have some carotenoid, but it's so little that it doesn't make an effect, that the chlorophyll overwhelms, okay, and makes it appear green. Okay, so make sure you understand that. Now here we have the action spectrum. So here we had the absorption spectrum showing which wavelengths are best absorbed by these specific photosynthetic pigments. Now the next slide shows you how good photosynthesis happens at each wavelength. So because most plants have these chlorophylls, at these wavelengths where these chlorophylls absorb a lot of wavelength, these areas will have a lot of photosynthesis. So the blue light will cause a lot of photosynthesis, and the red light will cause a lot of photosynthesis. When you only give them green light, that will not lead to any photosynthesis. So we can see here, photosynthesis happens the best at these lights, at these wavelengths, where the photosynthetic pigments absorb a lot of wavelength, and here. And where in the middle, where it doesn't absorb it at all, there's low photosynthesis. So make sure you know the difference between the absorption spectrum and the action spectrum. So I said a lot of stuff here, and they're summarized here in this table here, where I show you a little bit what's going on with the absorption spectrum here and then the action spectrum. Okay, so now we're going to another interesting thing. I find you probably guys will do this experiment in your class, maybe not, but I'm pretty sure you guys will. So this is a pretty interesting experiment, and what happens in this experiment is called chromatography. So this is a technique used to separate something into its constituents. So in this example, we're going to basically separate, we're going to take a plant, we're going to smush it, we're going to take that smush and put it here on the paper, okay, on the paper. And we're going to try and separate all the photosynthetic pigments that is in this smush, in this little mash. Because we know the plant has a lot of photosynthetic pigments, and we want to separate them out, okay, to see which pigments it's made of. So we can see here, this technique, basically we have a wooden rod here and a paper attached by a pin, and the paper is hanging here, and we dipped it in a glass. The glass has some water in here. We call that has a solvent in it. It could be water, it could be something else, depends what you want to use. So here's the solvent, basically the liquid, and we have here our stain that we put here. Now, once we dip the water into the solvent, the water will start creeping up and passing by the leaf stain. And depending on how soluble the pigment is, will influence how far it travels. So, those pigments most soluble in the solvent, so meaning likes the solvent, they will travel farther up the paper compared to those that are less soluble in the solvent. So different solvents, you can also use different solvents. So different solvents will result in a different separation outcome. Okay, so let me show you what will happen here. So you can see here, now we let the experiment go, the water started creeping up, and we can see we form a bunch of these lines. A bunch of these lines were formed as the water moved up. So we can see all the pigments. You can see carotene, this pigment loves water. Loves the solvent. It was very soluble in the solvent, so it traveled very high, all the way up. Where some of these other ones, for example, chlorophyll, was obviously not that soluble, so it didn't travel as far up this paper. It only traveled a little bit, so the carotene would be very soluble, and these chlorophylls were much less soluble. But you can see this technique is very cool, because we were able to separate out all of these pigments, these photosynthetic pigments, and see which ones exist, because maybe we don't know what this plant which pigments this plant has. So if we want to test it, we just do this experiment, and now we can see, this plant has chlorophyll A, chlorophyll B, xanthophyll, and carotene. It has all of these pigments. So this kind of technique will show you what pigments make up the leaf, make up what pigments are located inside the leaf cells. Now, it doesn't show you how much of each of these pigments there are, it only shows you which ones there are. So obviously I told you chlorophyll is the most common, their concentration is really, really high, and these other two would be really low. Okay, so it doesn't show you the concentrations, but it shows you which ones there are, okay? So now, I want you guys to try this question out yourself and see if you really understand this. So, this chromatogram, which is just this right here, presented was obtained in the laboratory. Calculate the RF value, stands for retardation factor, believe it or not. Retardation doesn't only mean you're mentally impaired, it also means retard means slow. So retard means slow. So it's a slow factor. So how quickly or how fast they move up, that's called the retardation factor. So we can calculate the retardation factor of a photosynthetic pigment just by a simple equation. The distance moved by the substance, so let's say carotene, over the distance moved by the solvent. So you can see the solvent moved 12 centimeters, the carotene moved 11.5. So it would just be 11.5 over 12. That would be your retardation factor. The higher the retardation factor, it means the further it moved, right? Because, let me show you here. Because 12, for example, 11.5 over 12 would be like 0.9 something, okay? It will be very high. So that's a very high, the highest retardation factor is 1 and the lowest is 0. The closer you are to 1, it means the further it moved. The further it moved, the closer it is to the solvent. Okay, so chlorophyll here would have a much lower retardation factor. It was much less soluble in the solvent. Okay, so you can calculate all of those and you can see the difference. It's very simple, but make sure you remember this. It can be asked. So now I have some other questions for you to see if you really understand it. So explain which of the pigments was most soluble in the solvent. Well, it's going to be carotene, because carotene has the highest retardation factor. It moved the furthest, meaning it was very attracted to this water. So when the water came by and moved up, it wanted to stick with the water, so it moved with it. It had some attachment issues, okay? Now these colors were not visible. So when you look at all, you can see all these different currents, orange, yellowy, green. So when we took our leaf, there was no yellow or orange. So why is there now? Like I said before, it depends on the concentration. So this leaf is made up of has all of these pigments, but it has way more of chlorophyll. So these are masked. They are almost overwhelmed that even though they're there, their properties aren't shown because they're overwhelmed by all of the chloroplast. So the yellow doesn't even show, or the orange doesn't even show. So it depends on the concentrations. Maybe there's another kind of plant, maybe that has a lot of these carotene and very little of these, and that one will appear orange. Okay, so it depends on the concentrations. Okay, now suggest why the calculated RF values are different to those in the chromatogram above. In a second, I just want to show you the answers written out because I said it, but I want to show you it as well. Okay, so here's the solution for written. Now let's do the last one. Why the calculated R values are different to those in the chromatogram above. So we know you can get different RF values depending on what solvent you use. Maybe this solvent is very good at moving carotene very far. Carotene loves this solvent, but maybe if you use another solvent, chlorophyll will love that solvent, and so chlorophyll will move very far. So the results you will get will depend on your solvent, okay? So a different solvent will show you a different result, so bear that in mind, okay? It's not going to be the same result with every solvent, so it's going to depend on what solvent you use. Okay, awesome. Okay, so that was a lot. I know this chromatography thing is not that interesting to you guys, but it's very important. It's super interesting what it can do. We've covered now so far, let's go recap, what photosynthesis is, why it's so important, difference between animals and plants with regards to photosynthesis and cell respiration. You need to know what autotroph and heterotroph is. You need to know the idea of light, okay, the absorption spectrum, these photosynthetic pigments, the absorption spectrum, the action spectrum, the differences between them. You need to know what chromatography's use is with regards to these photosynthetic pigments. And then now we're going to move to the easier parts. You need to know what factors affect photosynthesis. Okay, awesome. Oh yeah, by the way, while I'm thinking about it, remember that cell respiration has the exact opposite equation as this one. Okay, so it's going to be not carbon dioxide, water forms glucose and oxygen. It's going to be oxygen plus carbon plus glucose will form carbon dioxide and water. So they're the exact opposite. Okay, just in case you didn't know that. Okay, so what are the factors that affect photosynthesis? Light intensity, carbon dioxide, and temperature. Let's look at them. So here we have a lovely graph. You need to know how to recognize this. This is going to pop up and you're going to have to recognize it and be able to say something about it or even answer a multiple choice on it. So you can see on the x-axis, the increasing light intensity. So here, low intensity, medium, high light intensity. So essentially daylight. Early morning, you're going to have very low intensity, and then midday, you're going to have a high light intensity, right? And then the y-axis here, we have the photosynthesis. So we can see as light intensity increases, so does photosynthesis. But after a while, it starts plateauing. So even though at this point you're giving more light intensity, the rate of photosynthesis doesn't increase anymore. It stays relatively flat. Why is that? I want to give you this great analogy. So here we have people, just since COVID passed recently, here we have some testing people. We love them so much because it means that things are happening. And here we have Messi, okay? And he's coming and he's going to go do a test. There's 5 people to choose from. So he goes, he gets tested. So we know in this scenario, if it takes 1 minute to test one person, we know right now the maximum rate that can happen is 1 minute, one person per minute. Now let's say we have 2 people. And now in 1 minute, we managed to test 2 people. Okay, so 2 Messis were able to be tested. Now if we put 5 people, in 1 minute, how many people can we test? 5. Okay, 5. Bear in mind, this will make sense soon. Now, if we add another person, how many people in 1 minute can we test now? Still 5, because there's only 5 testers. Okay, how many people in 1 minute can we test now? Still 5. What about now? Still 5. So you can see, even though we're increasing, messy, the amount of people, because there are only a certain amount of testers, the rate of testing will not increase. There's only 5 people, so only 5 people can be tested per minute, despite how many people you have. So the people here represented these testing people here represents our thylakoids, the place that is absorbing this light. And the messy here represents our, messy here represents our light. Okay, and these represent our thylakoids that are absorbing the light. So if we come here to the picture, so we only have a certain amount of thylakoids. So that means we can only absorb a certain amount of light. If we give it more light, if all the thylakoids are busy, then more light wouldn't help because all the thylakoids are already busy. All these testing people are already busy. Giving them more light, more messies, will not make them go any faster, will not make them do photosynthesis anymore. That's why the rate slows down. So initially, as we increase light, photosynthesis increases and increases, and then eventually the thylakoids will get so busy, so full, that even if you give them more light, they can't go faster, and so it will plateau. Okay, so that basically, these guys here represent enzymes that are working in the thylakoids, right? In our thylakoids, we have the enzymes that are using this light to split apart the water, but they will get full. They won't be able to do any more. These photosynthetic pigments will be full. Okay, so here we can label it. Enzymes already working at their maximal rate. So I hope this analogy makes sense why it plateaus, because if you understand that, then you'll understand the next one as well. Carbon dioxide is another factor. We can see as we increase carbon dioxide concentration, photosynthesis will start increasing gradually and eventually plateau. Why is that? Because we know, let me bring this back, carbon dioxide is needed here for carbon fixation. Remember, we need to combine carbon dioxide with the hydrogens from here, okay, to make glucose. Now, if you increase carbon dioxide, that means you can make more glucose. But there's an extent. If there's an enzyme, remember, there's an enzyme that's combining these hydrogens and carbon and oxygens together to make glucose. Now, there's only a certain amount of enzymes. So if you give them more and more carbon dioxide, they will start being full. They will start working at their optimal rate. So giving them more carbon dioxide will not necessarily help because they're already working at their maximal effort. Okay, so make sure you understand that. Enzymes are already working at their maximal rate. Okay. Next one, temperature. So we can see from this graph, it's a bit different from the other two. So as we increase temperature, things happen here. So we know photosynthesis has a lot of reactions with a lot of enzymes, and we know each enzyme is just like a working man. Let me show you here. It's like Bob the Builder. It's going to build things. It's going to make reactions happen. Now, enzymes, okay, have an optimal temperature, okay? Just like us humans, when we sit outside, we have a certain temperature that's just too hot for us. We're too lazy, don't want to go do anything. Then there's temperatures that are too cold for us. We're too lazy, just want to stay inside. Enzymes are the same way. They have an optimal temperature. So you can see here, for photosynthesis, this temperature here, for example, is the optimal. Any less than this is too cold. Any more than this is too hot. Okay, so what does it mean? So when we increase the temperature, we know that's going to increase molecular collision. So when you have a lot of reactants like carbon dioxide, water, hydrogens, all that's doing photosynthesis, they move around at a certain speed depending on the temperature, these little reactants. Now, the higher the temperature, the faster they move, which means the more likely they are to collide with an enzyme, and therefore the more likely they are to form a product, which is glucose, okay? So the speed of these reactants moving around and colliding and making things happen is influenced by temperature. The more the temperature, the faster they move. The increased, the more the collisions there are, and the more products can be made, okay? But there's a limit to that. There's going to be a certain point where if you cross that point, you are no longer benefiting it. You are now causing these enzymes to become denatured. Remember, an enzyme is a protein. It's made up of amino acids that are linked together. At a certain temperature, if you pass a certain temperature, it becomes too hot and you break apart the bonds holding together this protein. So you start denaturing it, and that's basically breaking it apart. And if you're breaking it apart, now it can't function anymore. It cannot bind these reactants anymore to make the product like it used to. Okay, you're breaking its tertiary structure. So that's why. So make sure you know how to recognize the influence of carbon dioxide and light and temperature on the rate of photosynthesis. Make sure you can recognize this graph and vaguely explain it for a big question. Okay, awesome. Now let's go to this last bit here before we go on to our last segment. Okay, so you can see here that we have, for this first line, we have low carbon dioxide, cold temperature, and this is the rate of photosynthesis. Now, we see you can make a curve where you combine all of these factors. Okay, so you can see here it's a low photosynthesis, but now if we increase the temperature to a more suitable temperature, the rate of photosynthesis will increase. If we increase the carbon dioxide even more to the most it can be, the photosynthesis will increase. So make sure you can also recognize that all of these factors are together. We never just look at one at a time. They all influence photosynthesis together. Okay, so now we're going on to the last little bit where we have to talk about measuring photosynthesis. Then we're going to go on to some questions to test your understanding. So when you and your friend, okay, because you guys love plants, okay, trust me, you love plants. You and your friend go and you both buy a plant and you want to see, okay, let's see who can make their plant do photosynthesis the most, okay? And you're like, I bet my plant can because my plant's cooler. Now, how can you guys measure photosynthesis? How can you say, actually, my plant did more photosynthesis than yours? What are ways in which you can measure photosynthesis, the rate of photosynthesis? There are 3 ways: biomass, bubbles, and pH, okay? Let's start off with the first one, biomass. You and your friend can go and put your plant and make it grow. The bigger a plant, the more a plant increased in size, the more likely it did more photosynthesis, because we know photosynthesis is the process of making food so a plant can eat. So the more food it can make and the more it can eat, the bigger it's going to grow, right? That's biomass. Okay, so the more biomass a plant gained, bio just means living and mass just means weight. So the more living weight it gained, the more photosynthesis most likely happened. So that's one pretty simple way for you and your friend to decide which one did more photosynthesis. But the problem is it's not that accurate. Other than photosynthesis causing increase in size, there are also other things that can cause an increase in weight of a plant, right? For example, maybe there are some deposits of poop landing on the plant, and that's actually not contributing to the real weight. Okay, so no, there are other things besides photosynthesis that can cause a plant to be heavier. Maybe there's some calcium or some things depositing. Maybe there's a little organism sitting somewhere or whatever. So no, there are other things that can cause a plant to increase in size and become heavier. So that may be so maybe that would influence the real result. Okay, so biomass is not the best way because there are many things that can influence a plant's weight, not only photosynthesis. So let's look at the next one, bubbles. Remember from our equation, right? From photosynthesis makes what? Let me go show you. Photosynthesis makes oxygen and uses carbon dioxide, right? Both of these are little gases. So see here, these red ones would be oxygen that's being made, and these blue ones here would be carbon dioxide in the surrounding the plant. So we can measure the bubble. So if you put for example, it's very easy if it's an aquatic plant because then you can put it underwater. If it's not aquatic, this technique maybe doesn't work that great. But if you put an aquatic plant underwater and you watch and you look at it, you'll see bubbles will start forming. We know these bubbles can be oxygen or carbon dioxide, right? So the more bubbles there are, the more photosynthesis is being happening because that means the more oxygen is being made, okay, by the plant. So that's kind of an easy way to see photosynthesis. But remember, this is for aquatic plants because above the water, you can't see these bubbles forming. Okay, so that's a very simple way, but the better way would be using a machine called a photosynthesis. So O2 production occurs with photosynthesis. We can see with eyes as bubbles, but also with other more accurate methods. So for example, if you don't have an aquatic plant or you don't want to count the bubbles because that's not very accurate, you can use something called a photosynthesis. Let me show you. It looks vaguely like this. So you have a little beaker, you have water, you put your aquatic plant there in a little container, and you connect a pipe to it, to a little thing. to a little syringe here, and you basically, what you'll see is how much air is formed, because this pipe is full of water. Okay, this pipe is very much full of water. So when gas is starting to form, the water will be pressed up, will be pressed and moved, right? And then the gas will be under the water, moving the water along. So depending how much the water moved, that will show you how much gas formed, okay? How much oxygen formed. So this is a more accurate way than just counting bubbles with your eyes. You guys might use this when you actually do this in the lab. I'm not sure what your teacher will do with you guys, but this is called a photosynthemeter. And what it does is it measures much more accurately than counting bubbles the amount of oxygen produced. And you'll get much more used to how this works when you actually carry it out yourself. Just know that the photosynthemeter is much more accurate compared to just counting bubbles. Now, I want to highlight something very important. Remember how I told you that a plant does both photosynthesis and cell respiration. So we know photosynthesis is going to make oxygen, right? So we know we can measure the rate of photosynthesis by using this photosynthemeter to calculate how much oxygen it made, because the more oxygen means the more photosynthesis. Now there's a problem. We know plants also do cell respiration, so they will also in fact use oxygen. They not only make oxygen, but they also will use a little bit of it to do cell respiration, to break down glucose to make ATP so that they can survive. So that's kind of tricky now. Ooh, what does that mean now? So we know photosynthesis will allow them to make oxygen, but at the same time, they're using that oxygen. So how do we know that this amount that we're calculating here is truly accurate, right? Because not only do they make it, but they also use it. So the final amount that we're going to calculate with this photosynthemeter may not be that accurate. It may be less than the true amount, right? Because in reality, this plant is doing photosynthesis, but some of it will now be taken away by cell respiration. So the amount of oxygen that we're going to calculate is going to be less than the true amount that it should be from photosynthesis, right? So we can look at it in terms of this little graph here. So we can see during daylight, the plant is doing photosynthesis, a lot of oxygen is being made. Then at night, when there's no photosynthesis, let me explain it a bit better. So at any given moment, a plant cell respiration is constant. So the rate at which a plant uses oxygen is always constant, no matter day or night. It's always relatively constant. But photosynthesis is not constant. During the day, photosynthesis is happening, okay? But at night, it's basically zero, okay? So at this moment, cell respiration is constant, this moment it's constant, this moment it's constant, this moment it's constant. But photosynthesis is only good here, and there's nothing here, good here, nothing here, okay? So we know the amount of cell respiration that a plant does can be calculated right here, because no photosynthesis is happening, which means the amount of oxygen used here is purely due to cell respiration. So we can take this amount here, because we know this is the amount of oxygen taken away by cell respiration. And we know here, at this time, it's going to do the same amount. Okay, we can't see it because photosynthesis is also happening. But in reality, at any given moment, cell respiration is taking away this much oxygen. So if we want to find the true amount of oxygen made during photosynthesis, we need to have this amount here, and we need to add this amount here on top of that, because that amount here is being taken away from cell respiration. So this number here is not actually right. Oxygen being made during photosynthesis is actually more. It's this amount here plus this amount that is being taken away by cell respiration. So make sure when you're doing your calculations about photosynthesis, when you're doing a lab or something, remember cell respiration is also happening. So you need to adjust your values to take into consideration the oxygen being taken away by cell respiration and add that on to the amount that's being done for photosynthesis so that you get the true number. So measuring the rate of oxygen production or carbon dioxide intake allows direct measurement of photosynthetic rate. If correction is made, you need to make the correction. You can't just say, oh, this is photosynthesis, because it's not true. More is actually done because cell respiration is taking away some of it. Okay, I hope this idea makes sense. It's a little bit tricky. If you don't get it, don't worry too much about it. It's a very small part you need to know, but try to get it and try to understand that you need to compensate, okay? Because cell respiration is also affecting the amount of oxygen, not just photosynthesis, okay? Awesome. Now the last one we need to talk about here is pH, okay? So we know that the surrounding water has carbon dioxide and water. Now carbon dioxide, okay, so this one here, when it dissolves in water, it forms something called carbonic acid. So acidic, it's very acidic, okay? Now we know photosynthesis. What does photosynthesis do again? Let me show you. Photosynthesis is going to use carbon dioxide from the environment. It's going to take away carbon dioxide from the environment, okay, to make oxygen. So it's going to remove carbon dioxide from the environment, okay? That's very important. So this plant here, when it does photosynthesis, it's going to take away this carbon dioxide. It's going to remove it from the environment. So I just told you that carbon dioxide, when dissolved in water, is acidic. So if this plant keeps on removing carbon dioxide, what does that mean in terms of the pH surrounding in the water? So if there's a lot of carbon dioxide, the pH is very low because it's acidic. But if this plant keeps on removing the carbon dioxide, it's going to make the surrounding pH higher because it's removing carbon dioxide, and carbon dioxide is acidic. So if you remove this acidic thing, the surrounding area is going to get more basic, more alkaline. The pH will go up, okay? So CO2 is used up in water in photosynthesis. CO2 in water is acidic, carbonic acid. So hence, the more photosynthesis, the higher the pH. So if you stick a pH probe in there, and you can also see that the pH will start increasing as photosynthesis increases. Let me show you here. Remember, acidic has low pH and basic is high pH. So to answer the question, when you and your friend compete and you're trying to see who did more photosynthesis, whose plant did more photosynthesis, an okay way to use would be biomass, but it's not that accurate because there are many things affecting the weight of a plant. Another thing would be bubbles. This is one of the best ways because you can directly use a photosyntometer, which can exactly calculate the amount of oxygen made. But remember to compensate for cell respiration if you want to find the true amount of oxygen made by photosynthesis. So in my opinion, this would be the best way, bubbles, okay, using a photosyntometer. But pH is also a good way. You can measure the pH as it's changing, as the plant is doing photosynthesis. It should get more basic as the plant is doing more photosynthesis. But this is an indirect way. It's not directly measuring one of these things. So the pH is not directly measuring something that's made from photosynthesis like oxygen or glucose. It's measuring a consequence of photosynthesis. So it's an indirect way. So it's technically not as good. So know all of these three ways. They're very important. Make sure you understand how they work. Know that bubbles is very, very good way. The photosyntometer, these two are okay, but not as great. Okay. So now that's it. That's it for photosynthesis. That's all you need to know. You need to know the factors affecting photosynthesis. You need to know how to measure photosynthesis. You need to know how photosynthesis works, the basic steps, and you need to know about the absorption and the emission spectrum and chromatography. So there's a lot of stuff, but I think you guys got it nailed. I hope you guys understood it. Now let's do some questions. So the image shows an electron micrograph of mesophyll cells. What is the name of the structure labeled X? So we can see here, this looks a lot like the image I showed you previously. The whole water inside here squeezing all the organelles to the side. So we know these are probably going to be chloroplasts. It's probably going to be D. But I want to explain something quickly. When you hear the word mesophyll cell, mesophyll is plant. Okay, it refers to the inside of a plant. Okay, to the inside of a leaf. I mean, okay, to the inside of a leaf, a plant. So you should know from this already that we're talking about a plant cell, not an animal cell. Okay, therefore you could know that this is going to be a plant cell. Therefore, you know a chloroplast is possible. So it's going to be chloroplast. Okay, if you look closely, you can see the basic structure. How can the rate of photosynthesis be measured? By the amount of oxygen produced? Yes. Remember, photosynthesis makes oxygen as a byproduct. So you can measure the oxygen, therefore you can know the rate. By the increase in biomass? Yes, you can. It's just not such a good way, but you can. By the amount of CO2 produced? No. Photosynthesis does not produce CO2, it uses CO2. So it cannot be this one. So it's going to be 1 and 2. So the answer will be B. What is the light energy used for in photosynthesis? To absorb carbon dioxide? No. To split water molecules. Yes, remember the light energy will split the water molecules in the thylakoid. Okay, cool. It's going to be B. How does chlorophyll, remember the photosynthetic pigment, respond to red, green, and blue wavelength in the white light? Okay, remember it reflects green light, so we can already look for that. A, B, and D are already showing reflect, so it's going to be A, B, or D. And we know it likes to absorb red and blue light, so it's going to be D. Okay, because red is absorbed, green is reflected, blue is absorbed. Okay, one more multiple choice question here. What are final products of photosynthesis and aerobic respiration? I want to show you this one because they like to compare these two because they're opposite processes, okay? So we know from this video that photosynthesis makes what? Makes glucose and oxygen. Okay, aerobic respiration will make the opposite things. It will make carbon dioxide, it will make water, and it will make ATP. Okay, the answer is going to be B. Make sure you know the difference between these two. Okay, now here as promised is a big question. Oh, sorry. A big question. The screen kind of cut a bit, so I can't move it right now. Let me try and do this for you guys. Here. Okay, so here's a big question. So I explained the process of photosynthesis very early in the video. Here is a way that you can figure out how to answer it in a question. So outline how light energy is used and how organic molecules, so glucose, are made in photosynthesis. They always ask in such a confusing way, but they're just asking you to describe photosynthesis, okay? So the question can be various amounts of points, from 4 points, 9 points, whatever. Let's say they ask you a very big question here, 9 points. Here are some things you can write. I added a lot of these, okay, because for the new syllabus. So make sure understand all of these little points. I explained them in the video. If you can just list 9 of these, you'll get full marks. So there are a lot of points here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. There are 13 things you can write here. You only need 9 of these to get full marks. So 9 of these will give you the full marks. So that's already interesting. You don't need to know how to write all of them, but make sure you understand them and try and remember as many as possible so you can answer a big question like this one. So that's it for this video. I really hope it was useful. I know there's a lot of stuff, but photosynthesis is a high yield topic and a lot of questions can be asked from it. So see you in the next one.